All right, the electron transport chain. I do have a note from yesterday. So yesterday was all about the citric acid cycle, right? And I called it the citric acid cycle the entire class. Because it has multiple names, I just kind of latched onto one. The citric acid cycle is also called the Krebs cycle. I tend to go with citric acid cycle because I tend to get Krebs cycle and Calvin cycle mixed up, and the Calvin cycle is in photosynthesis. So if you want to flip to your notes from yesterday, and next to citric acid cycle, write Krebs cycle, Krebs cycle. That way you like have the all of the vocabulary for it. It's just named after the guy who invented it or discovered it or named it or whatever. But that is the Krebs cycle, the citric acid cycle. Those names are interchangeable. I just get the C sounds mixed up, so I just went with citric acid cycle. Does anybody have any questions about that stuff before we move on to electron transport chains? Krebs cycle, it is. Find a darker marker. Same thing, different names. Scientists like to name things a bunch of times because nothing can be that easy, right? But today we're talking about the electron transport chain, also known as oxidative phosphorylation. We'll get to that in a moment. My goal for you is for you to be able to describe how electrons in the citric acid cycle, the electrons that we got from our Krebs cycle, are coupled or put into ATP via chemiosmosis. So, big, strong words today, but relatively simple concepts once we get them. So, starting off, where are our electrons, right? We finished off yesterday, and we had NADH and FADH2. Those alphabet soups are electron carriers. We got some from glycolysis, and we got some from the oxidation of pyruvate, and we got some from the citric acid cycle or the Krebs cycle. And those two electron carriers are going to be shuttled to the electron transport chain. The electron transport chain is in the inner mitochondrial matrix, or in the inner mitochondrial membrane, sorry. So if we've got a mitochondria, remember that mitochondria has two membranes, right? The electron transport chain is embedded in this internal membrane. It is embedded in this internal membrane. That is where our electron transport chain is. Because we really need this compartmentalization, and we'll talk about why in a moment. You don't need the chemical structures of NADH and FADH2, but I just kind of threw them up there to show you what we're working with. There are very subtle differences between the two. Obviously, with NAD plus and NADH, we've added an H just to this top little ring, and with our FADH2, we've added two hydrogens to different nitrate groups in the molecule. So these function as our electron carriers. This is what we're bringing in. We're done with the carbons. We're done with the carbon dioxides. We're done with our We're done with NAD plus and FAD. We just want our energized electron carriers. These are our energized electron carriers. These have our energy. So what is our electron transport chain? It is, I misspelled inner really, really badly. It is a collection of molecules, a collection of proteins in the inner mitochondrial membrane. It was so bad. Why is my caps lock on? There we go. Inner mitochondrial membrane in the mitochondria of eukaryotic cells. Eukaryotic cells aren't the sole perpetrators of cell respiration. Prokaryotes can do that too, but prokaryotes don't have internal membrane systems. So their electron transport chain is on the outside of their cell membrane. It's just on the outside of the cell. We have 4 steps in the electron transport chain. They are proteins numbered 1 through 4, but we use Roman numerals. And there are a couple coenzymes and cofactors that help it along. So it's a bunch of proteins embedded in this membrane. There are 4 of them total. Again, inner mitochondrial membrane, not the membrane of the mitochondria, but the membrane that is inside of the mitochondria. So just like any other protein that we've seen embedded in a membrane, it's just this specific membrane and those specific proteins in that 1 through 4 order. So that's what we are referring to when we talk about the electron transport chain. It's these 4 little proteins in this mitochondrial membrane. All right. All right, so this is what it looks like, and we're going to draw this together slowly but surely. So we've got our membrane. So we're going to flip back and forth just to make sure we have it. So we've got, no, we want this and this. Yes, please. Okay, so we're going to start out with a membrane. Why did I do that? We're going to start out with a membrane, and we're going to make sure we label it the inner mitochondrial membrane. We've got our membrane. It is a bilayer. I don't want to draw out the individual lipids. That would take a very long time. So we're just going to represent it with these 2 little lines. So the outside of this is going to be our inner membrane space. So the space between the external, like the membrane of the mitochondria, and on the inside of that inner mitochondrial membrane is the mitochondrial matrix. So that'll kind of locate us inside the cell. We've got 4 proteins, the first of which is a transmembrane protein. So it's going to be on both sides of the cell, and that is going to be complex 1. We use Roman numerals to differentiate between these. This is complex 1. Flipping back to our more detailed image, right? We then have complex 2, which is not a transmembrane protein. It doesn't go all the way across. So we have a little bit of space in our membrane, and we have complex 2. Before we hit complex 3, there's a cofactor that is used to help. We call that cofactor Q. So we can have a little co-Q up here. And this is the order that those electron carriers will go through as they move down the electron carrier or electron transport chain. After CoQ, we have complex 3, which is another transmembrane protein. Ooh, that works. Complex 3. We have another cofactor to help. That is cytochrome C, or CYTC. And another transmembrane protein gives us complex 4. And that is our electron transport chain. At the end of our electron transport chain, we have a very special protein. That special protein is shaped a little bit differently. We've got a transmembrane kind of component, and then it actually has a spinnable part at the bottom. So usually it is drawn like a little pumpkin. I'm going to try to do that without getting... There we go. Cool. This is ATP synthase. We'll talk about why ATP synthase is so important in a minute. This is where the magic happens. This is how we get most of our ATP from our glucose. This is the stage on which most of cell respiration happens, where the meat of cell respiration happens. Doing okay so far? All right, so here we go. Let's simplify this down for a little bit. This sentence right here is our kind of overview, our main idea. Our electrons are going to move from a less electronegative carrier to a more electronegative carrier down the chain. So complex 2 is more electronegative than complex 3, which is more electronegative, or which is less electronegative than complex 4. So we're moving more and more and more electronegative as we go down the line. And if we wanted to add that to our little our picture, we've got electronegativity increases as we go down the line. Went too far. So, when these are transferred between molecules, an electrochemical gradient of hydrogen ions is established. As these energy electrons are moved through this electron transport chain, it uses the energy that is released to pump hydrogen ions across the inner mitochondrial membrane, and that is how we establish our electrochemical gradient. And this is what is used to generate ATP. This is our second to last step. If our last step is make ATP, our second to last step is establishing this electrochemical gradient. This is our overview of the electron transport chain. This is the big idea for our electron transport chain. We pump hydrogen ions, we establish this electrochemical gradient, and we use that electrochemical gradient to generate ATP. And you only have to write what's underlined. So that's our idea. Our electrons on our carriers will hop from complex to complex to complex, getting more and more electronegative as they go, meaning that energy is being released. Yes? I can. Yeah, we'll probably flip back to the drawing a couple more times too. There we go. So, how on earth are we going to use an electrochemical gradient to generate ATP? Well, first and foremost, do we remember what an electrochemical gradient is? Maybe a little bit. When we talked about membrane potential, right? And membrane transport, we talked about how ions like to move, or things like to diffuse naturally from areas of high concentration to areas of low concentration, right? An electrochemical gradient is that difference between high and low. So if we've got a lot of stuff on one side and not a lot of stuff on the other, we can consider that an electrochemical gradient, especially if these have charge. So if we have H plus ions, like we do, electro, because we've got the plus, chemical, because it's hydrogen, and there's a gradient across the membrane. That's what we are establishing. We've got a difference in the hydrogen ions on either side of this membrane. And there's a special protein that helps us turn this difference into ATP, and we call that ATP synthase. ACE means it is a enzyme. And enzyme names tell us what they do. ATP synthase synthesizes ATP. It tells you what it does in the name. So it'll take in ADP and inorganic phosphates, those little PIs that we draw. It'll take those in and it will make ATP out of them. It will trap that energy, it'll make that bond. Essentially, it works like an ion pump in reverse. So, in our little diagram, we're going to have lots of H plus up here. And a little H plus down here. It works like an ion pump running in reverse. So ion pumps will pump ions against their concentration gradient. This allows ions to diffuse down their concentration gradient. It will use the energy difference between our two sides to generate ATP. It's actually really stinking cool. Because these hydrogen ions, do they want to go up or down? Are hydrogen ions going to go up, or are they going to try to go down? Based on down, right? There's a lot up here. They want to space out, so they want to go to where there's less of them. So physics will have these hydrogen ions wanting to go through this portal. And ATP synthase lets them through, but it uses the physical force of these hydrogen ions moving through this to spin this bottom part, and that spinning allows ATP to be synthesized, which is really cool. This stuff's just like moving. But this little protein is the reason we can make ATP out of our food. So, we're using the difference in those hydrogen ions to generate ATP. So, we have a special name for this because we have a special name for everything, and we call it chemiosmosis. Chemiosmosis. So, when we use energy stored in that gradient, that is what we call chemiosmosis. We're using an electrochemical gradient to our advantage. We're using it to do work, to power ATP synthesis. And when we do that, we call it chemiosmosis, which is just a fun word to say. I recommend trying it out. Our difference in hydrogen ions creates our force. So if you wanted to do a little doodle, nothing too crazy, we've got our ATP synthase in here. Our ATP synthase. We're going to have a lot of hydrogen ions out here. If you can even see that. And what happens is those hydrogen ions are going to go through that ATP synthase, and we're going to go from ADP and our inorganic phosphates, we're going to make ATP out of it. We are using this gradient to our advantage, and we call that chemiosmosis. Doing all right? Okay. So, we're doing the work. We are generating ATP. Everything we've done from glycolysis to the oxidation of pyruvate to the Krebs cycle was all about generating those electron carriers. Those electron carriers are used to establish this gradient, and that gradient drives ATP synthesis. So, how are we actually building that gradient? We've talked about what is building the gradient. We've talked about what the gradient does, but we haven't talked about how it's established. Our flow of electrons from NADH and FADH2 is exergonic, which means it releases energy. As those electron carriers are passed through our electron transport chain, they are releasing energy. That release of energy is used to pump hydrogen ions across the membrane out of that inner mitochondrial matrix into the inner mitochondrial space. ATP synthase is the only way for those hydrogen ions to go where they want to go down that concentration gradient, to do what physics is driving it to do always. This creates something called proton motive force. Bless you. When I really got into the weeds of what proton motive force is, I just did it. I was like in a college bio course. We just need to know what proton motive force is, but if you take a higher level bio course, you get into it more. And my professor had a really heavy accent, so he would push these words together and call it protomotive force. So every once in a while, protomotive force will sneak out because that's the way I was taught about it. But it's proton motive force. It just gets going really fast. That force, because when things move, it has a force to it, right? If you've ever been in a wave pool, that water moving pushes you backwards, right? Those hydrogen ions moving has a force with it. That force is called proton motive force because our protons are moving and it's creating a force. And that force is what's going to generate ATP. Like cranking a turbine in a wind field. But instead of electricity, we get ATP. Doing okay so far? So we're just pumping these out in order to harness the energy of them moving back in. So this is how it works. And we're going to draw a very simplified version. We're not going to draw all those little phospholipids. That would take a really long time. So this is our cytoplasm of our cell. This is our inner mitochondrial membrane. We have our intermembrane space, our electron carriers, or in our electron transport chain that we've already drawn. And we have our ATP synthase. So, let's do a quick doodle. Nothing too crazy. We're just going to simplify this. That way we have it. So, we've got our mitochondrial membrane. So, this is going to be our cytoplasm. We have our intermembrane space. And we've got our inner mitochondrial membrane. Ooh, going too quick. Does everybody have that kind of orientation? I know seeing things in 3D when things are 2D can get wonky. Everybody know where we are in our cell? If not, right here, we could literally take this cross section and it'd be that. So, quick electron transport chain. Right? Quick electron transport chain. Probably could have drawn that bigger, but c'est la vie. And we've got our ATP synthase. So this is our electron transport chain. And we're going to have our ATP synthase. Oop. So what's going to happen is we are going to harness the use of these electron carriers to pump out hydrogen ions. So if we have an NADH, it's going to be passed from electron carrier to electron carrier, right? We're going to get NAD plus out of it, and we're going to pump that hydrogen out. Same with our FADH2. We're going to go oxidize it to FADH, and we're going to pump those hydrogen ions out. At the very end of this, we've got oxygen. This is super important. This is where our water comes into play. We're going to take the last tiny crumbs of energy left in those electrons, and there is only one thing that our bodies would like to accept that electron. Our final electron acceptor is oxygen, and when we have those electrons get to that oxygen, that's where we get our water. That was the last step in our equation when we drew out the end of cell respiration yesterday. This is where the water comes from, because our oxygen is the final electron acceptor, and it's a very, very good electron acceptor. So we've established this electrochemical gradient. There's some hydrogens in here. That's what we use to make that water. But these hydrogens want to create a balance. They want to even stuff out. And when they do, actually, I'm going to move my ATP synthase down a little bit, just for clarity's sake. Oops. ATP synthase. These hydrogen ions are going to want to go straight through that only door in, their only chance to create balance. And when that happens, we're going to get from ADP and inorganic phosphate to ATP. I ran out of time today. We will pick this back up tomorrow. I will save this image or redraw it. We will have time to finish it up tomorrow. No worries. I'll see you then.